Hey guys! So, unfortunately I have an appointment this morning that couldn't be moved to any other time, and we don't have time because we're doing so many labs next week to push our notes back any farther, so we're just gonna have to do notes this way. I'm sorry, it's not how I like to do it. I like to be there and see your beautiful faces. Also, hopefully I'm gonna be able to keep this short, sweet, to the point. I don't know about that. But let's go ahead and get started. I'm going to minimize my face. There we go. Alright, so we left off yesterday talking about lysosomes, and we're going to pick up today talking about vacuoles. So our primary function for a vacuole is going to be to store stuff, whether it is water, whether it is food or excess macromolecules. A lot of animal vacuoles are used to store hormones. You can store minerals or ions, basically just anything that you need to put in a bag, you're going to put it in a vacuole. They're exceptionally important in plants, which is what this big boy here is with the big black arrow pointing to it. So that's called the central vacuole, and we'll talk about that in just a second. But vacuoles are membrane-bound vesicles, and again, exceptionally important in plants. I'm not entirely sure at what point a vesicle becomes a vacuole, because they're kind of the same thing. Vesicles are going to be short-term, moving around. I guess vacuoles are like we beef up the membrane itself to make it be more long-term as opposed to a short-term thing. Whatever. So in plants, vacuoles are exceptionally important because they help the plant maintain turgor pressure. And we'll talk a little bit more about that, I think, on Friday, and definitely next week when we talk about how water moves in and out of cells. But if you ever forget to water your plant, you'll notice that it will start to kind of sag and fall over. That is because this giant central vacuole in the plant helps the plant maintain its rigidity. So it has a cell wall, and it has this big water-filled bag on the inside that pushes out against that cell wall and makes it really rigid. And that's what helps plants stand up. When you don't water them, they kind of start to fall over, and it's because at that bend, the turgor pressure is too low, and so the cell wall is kind of sagging in on itself. We'll talk some more about that next week. So we have this GIF from yesterday. We're looking at this vesicle here. If we were to just keep it, it would be a food vacuole. We form these guys up whenever we do phagocytosis, and a lot of times you will fuse in a lysosome there for digestion. You can also just keep it in that vacuole and store it there for a while. Again, we're kind of using the term vesicle and vacuole interchangeably here because they're functionally the same thing. Alright, just for funsies, we also have these things called contractile vacuoles. They're very important in paramecia, like protists, so we got this paramecia protist down here. So you can see he's going to suck water out of the interior into that vacuole and then blow it out the outside. And so that's going to help him maintain water balance and maintain his salt concentrations in these single-celled organisms. So you maintain your water balance and salt concentrations by peeing and drinking water. These guys are too small to do that, so this is functionally how they do that. And I just think it's neat. You can see it literally having all of these cytoskeletal components that are, moving it in and out. It's cool. All right. And then our final thing that we're going to talk about with vacuoles is that central vacuole in the plants is also there, stores water, but it also can store what are called secondary metabolites. So plants make a lot of things that are not directly related to photosynthesis and carbohydrates and eating, like pigments that make it be colorful, like chemicals that give it weird traits. So these are tobacco leaves. The nicotine in those leaves is a secondary metabolite that doesn't help the plant grow, but is being produced either for defense or reproduction or stuff like that. Hi, sorry for the jump cut. The janitor came in and scared the crap out of me. So, yes, let's move on. So now we're going to talk about mitochondria and other plastids. And remember, earlier in this unit, we talked about how like mitochondria and chloroplasts used to be free-living cells on their own. So that means that they have some really interesting traits, like the double membrane, like their own DNA, etc. And so that's why we're kind of grouping these guys together. So, mitochondria, everybody remembers from, on-level bio that it is the powerhouse of the cell. Okay. However, we need to get a little more specific with that. So, it is the site of cellular respiration. Okay, that is where you take oxygen from the air that you breathe and you use it to break down carbohydrates into ATP. Okay, hopefully you remember ATP is the biological energy source. And that's how, our body converts everything back to ATP. This is what we use for energy. So we're not actually using the carbohydrates or the proteins or the fats for energy. We're using them to make ATP, which is our energy source. And it's how we can have, carbs and fats and proteins and all these different things that we eat still give us the same kind of energy, is because we're converting it back to our energy itself. I like to explain it to my on-level kids like, you can go to all these different countries and buy all different kinds of things with the money that they use over there, but you can always convert it back to the US dollar. So it's the same thing here. You have all these different kinds of things that actually have energy in them, but you convert them back to the biological energy unit that we use. Anyway, so mitochondria is the site of catabolism or catabolism. I'm not sure how you want to say that. Both are correct. But it is where you break stuff down. So that's part of your metabolic processes, where you are breaking bigger molecules down into smaller things. So we also call this aerobic respiration because you have to have oxygen to do it, as opposed to anaerobic respiration where you don't need oxygen. And remember from earlier, the mitochondria has a double membrane because it used to be its own thing. And this inner membrane is very convoluted. It's very like confusing and rippled, and it's bigger, and then it got squished and folded down into the smaller inner membrane. And again, surface area. It gives us all this extra room to do these chemical reactions in. Ah, beans. Why two membranes? What a good question. Surface area on that inner membrane, surface area on that outer membrane, and because it used to be its own thing, so this is the bacterial membrane, and this is the ancestral eukaryotic cell membrane. Okay, move me back over here. All right. So, we do some chemical reactions on that outer membrane. We'll talk more about that when we get to the unit where we talk about metabolism, but there is a semi-liquid like cytosol space in here. It's called the intermembrane space. That's where we're doing chemical reactions as well. My light just went out. Okay, cool. So in this inner membrane here and across it specifically, that is where we are doing the electron transport chain portion of aerobic respiration, of cellular respiration. Hopefully you remember from on level, there's a couple different stages to respiration. You have glycolysis, which occurs on the outside of the actual mitochondria, so out here in the cytosol. And then you have the Krebs cycle, which occurs in this inner matrix. So we move everything into the very inside, and then we just kind of let it flow back out. And as it flows back out, we're doing chemical reactions across these membranes, establishing chemical gradients and blah, 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 blah. But ultimately to generate ATP. So, now let's talk about the chloroplast. It's very similar to the mitochondria. Again, we have this double membrane. It has a special job. It has all this inner stuff. So, the primary function for chloroplasts, photosynthesis, duh, they're green. Okay. And I said plant-ish only because there are some non-plants that have chloroplasts. They're going to be your protists, or your plant-like protists, and some weird little like single-celled things will have, chloroplast elements in them, but that's even higher than us, so we're not going to get into it. So the reason that they're green and the reason that they are able to do photosynthesis is that they contain chlorophyll, which is the chemical in these thylakoids that makes it green and that allows it to capture solar energy. So again, we have two membranes here because we're trying to maximize surface area. And this outer membrane is the ancestral eukaryote and the inner membrane is the ancestral chloroplast bacteria plant thingy. Now, inside here we have all these little sacks, and it looks a lot like a stack of pennies, just like a whole bunch of stacks of pennies. You can see over here, like, it's just stacks and stacks and stacks. And so why do we have so many of those stacks, and why do we have, you know, all of these membranes? Again, it's to maximize surface area because we're doing chemical reactions across them. Now, there are some other plastids in plants, just for fun, not that you need to know. There are these things called amyloplasts, and they are for storage. They store starch in the roots of plants, so like potatoes have a lot of amyloplasts in their cells. Then you also have chromatoplasts, which are storing pigments, so in places like fruits and flowers. Again, these are secondary metabolites that are being stored there, so you can either store them in a vacuole or you can store them in these chromoplasts. It's kind of, you know, some plants do it one way, some plants do it the other. All right. So, yes. Okay. So you can see here, there are, you know, little, what I'm assuming used to be ribosomes. Yes, little ribosomes in there, because again, this used to be its own thing. There is DNA components in here as well. And this inner portion here is filled with a liquid, which I'm assuming used to be the ancestral bacteria's cytoplasm. Now we call it the stroma, because it's not chemically identical to, you know, the cytoplasm that we have, again, because it's not us, it used to be a bacteria. But in this stroma, in this inner fluid, that is where we do the Calvin cycle, which is one of the parts of the multi-step photosynthesis. So you have light reactions and you have Calvin cycle. The light reactions are happening inside these thylakoids, because that's what's capturing the solar energy. And then in the stroma is where you are doing the Calvin cycle, which actually makes the sugars, and then they're exported out of the chloroplast. These big stacks as a unit, the independent things are thylakoids, and then once you get them in a stack and kind of bagged together, we call that a grana or granum. And so, sorry, there's like a shadow. Spooky ghosts. It's literally my shadow. I hate myself. Okay, keep going. All right. So now I want to talk about peroxisomes, and I promise we're getting towards the end. Peroxisomes' primary function is to break down fatty acids into sugars. So they kind of help with lysosome duties, so they break stuff apart. They also help to detox alcohols and other toxins. So, like, you have lots of peroxisomes in your liver and, you know, in your kidneys and stuff like that. So peroxisomes, again, it's just going to be a membrane-bound thing. It's very similar to a lysosome. The interesting thing about peroxisomes, though, is that they have this giant crystalline core, which actually has like metals in it, and it works like a catalyst core. So you literally will take all of these chemicals and you kind of drop them into this crystalline core. The metals and the ions pull apart things, or they will, you know, chemically change. Very similar to like kind of how they do it in rockets. Like they'll drip stuff over an iridium catalyst to create the hydrogen that they use to fuel rockets. That's pretty much exactly what's happening here. You're dripping these chemicals across this crystalline core to break it apart into its base components. This is how you're doing the chemical reactions in your cells. Super neat. So in animals, we are creating hydrogen peroxide from all of those leftovers from respiration. So you have lots of oxygens, lots of hydrogens that are just kind of left over. And so it will, you know, they snap into hydrogen peroxide, which is not great. It destroys cells. So peroxisomes are in charge of breaking that apart into hydrogens and waters. And in plants, a lot of times they can convert oils, these fatty acids, convert them down into something more usable, especially in the seeds. So Plant puts a lot of oils into the seeds because it's easier to store that way, and then peroxisomes help break it apart so that the plant can actually use it. All right, I'm going to stop here and we will pick up in another video. I'm sorry, I promise we only have 1 or 2 more to talk about. I know this is very long. I'm very, very sorry. See you in the next video.